Hi everyone. I hope I'm audible. Let me just check once. Alright. Okay. So before I start, let me introduce myself. I'm Vani Kari, biology educator at Unacademy. I'm an engineer with bachelor's and master's degree in biotechnology and a B.Ed degree. Also a founder of a startup where I grow microgreens by vertical farming technology. So today we are going to discuss about the seed structure. Okay, so seed structure, so mainly we'll discuss about the monocot seed, all right, and then the dicot seed. So structure, like how exactly is the arrangement of different layers that are there, or the cotyledon, endosperm, then the main embryo. Okay, so we'll discuss about all that and what is the difference, we'll see. All right, so before I continue, so join with us in Telegram for homework and class notes. You can join the channel Vani Kari ICSE Class 9 and 10 biology it is. Okay, and also Unacademy ICSE and IHC both. So this link you can find in the description box. So go to the description box, click on this link, and then you can join this channel for updates regarding the classes. So also we conduct special classes on Unacademy platform, so that is basically free classes. Okay, so you can learn from your favorite educator for free, so that is engage with educator via poll. So this is basically we have an option to conduct a quiz there, so you have an option to choose between the options, and basically we'll be sharing the leaderboard so that you will know your rank and compete with your friends. So like basically I have a practice quiz that is going on right now. Okay, so you can just take part in those. So all you need to do is download an Academy Learner app, and take part in these practice quiz. Okay, so this will help you in preparation for your exams as well. Okay, so clear your doubts in the live special classes. So that is basically so here there is a question tab separately, all right, so that is during a live session. So in that case, any doubt you have, so you can type in your question. You have an option to attach an image and send it to the educator during a live session, so that is using a question tab. So as the educator is getting a notification, will be clearing your doubts during a live session itself. Okay, so this is to attend the special classes, all you need to do is download the Unacademy Learners app. Okay, so download it and start taking the classes. So you can choose your category as ICSE and start taking up classes. All right, so now for top special classes, you can go to the description box. There you will find some of the links. But anyway, once you download the Unacademy Learner app, you will understand how to go about this. So this is about the Unacademy subscription feature, which we call as plus classes. Okay, so these are plus classes. These are special classes, these are plus classes. The difference is that here only certain percentage of topics will be covered, and these are free classes. Here complete syllabus will be covered, so you can see learn live from the comfort of your house. So that is there. Unlimited access to all the courses, so different educators will come up with different courses, so you will have access to all the courses once you take the subscription. So this option is not available for special learners, because these are regular classes that are conducted. Okay, then top educators of India are going to take the classes, so that is basically Unacademy platform will be conducting the classes. So then there is a regular doubt clearing session and an answer writing session. So what is that doubt clearing session? So basically after about every 3 sessions, so you have one session which is completely dedicated for doubt clearing. So whatever doubts you have related to the topics that are previously covered, so you can clear your doubt in this session. So that is a doubt clearing session. Okay, so any doubt basically regarding the topics that have been covered in these three, so all of you can ask the doubt in this. So let's say you do not have any doubt, so we can conduct a quiz, or we can discuss some diagram based question type of questions, whatever are important, like mainly concentrating on previous year question papers also from that particular chapter. So we'll be concentrating in this particular class. So that is a doubt clearing session. Alright, then complete syllabus will be covered. So since it is a plus class, so it is 100% syllabus will be covered. Mentorship and guidance is given. Study material is also given, so that we upload in the form of a PDF. So you can download the same PDF from the session that you are attending. Alright, then there is practice test which will be given. Live test series are given. The batch courses will be there, like we have a batch in English, we have a batch in English. So now depending on your convenience, you can go with any of these batches. Okay, so now some prefer to learn in English, some prefer to learn in Hindi as well as English. So this is completely your choice. So once you take the subscription, so you will have access to the English batch also, English batch also. So you can go with any of the interest of your batches. Okay, and with that there is daily practice section, weekly mock test series, live quizzes are there, daily MCQ and subjective test series are also there. So you can go ahead with this. All right, so now since this is the very beginning, even though it is a June month and the academic year starts with the June month itself, so if you are planning to take the subscription, so you can since it is for class 9, so you can plan for class 9 as well as for class 10, or otherwise you can just try with 12 months or maybe 9 months, like 9 months. This is completely depending on your convenience. Okay, so in that case you can just have a look here. So for 12 months it is about 17,250, but with the referral code, that is Vani YT. So here you can see a gift box, so there you need to add a referral code Vani YT, where you will be getting a 10% discount on the subscription. So that is about by using this code. So it is about 17,250. On that you will be getting 10% discount on the subscription. So this one I just told you for 12 months. So you can also go ahead, like 9 months, or depending on your convenience, maybe you can plan for 18 or 24 months as well. So this is completely your choice. Alright, so let's get started with the seed. So we'll see what exactly is seed, what all does a seed contain. Okay, so now when we mention about a seed, what all is present, so how is it helpful for a plant growth, or why there is a need for a seed production, or we'll see whether the seed is a living thing. Yes, anybody has any idea on this one? So is a seed living thing? Is it having any DNA, or how do we use these seeds basically? So in that case we'll discuss all of this, like what exactly is seed. So seed structure we'll see in general, and then we'll discuss about the specific parts related to the seed. Okay, so now seed structure. So you can see that there is a seed coat. What is this seed coat forming the testa and the tegmen? Any idea? This one, only this part. So we are concentrating only on this part now. So now you might have studied already the fertilization. So if not, I'll just explain you what exactly happens. So after the fertilization process, okay, so that is fusion of male and a female gamete occurs. So you can see lot of changes that are gonna occur in a plant body. So mainly the flowers, when we mention, so petals are gonna fall off, sepals are gonna fall off. Then you can see basically the fusion of male gamete and a female gamete, then fusion of polar nuclei and another male gamete. So in all those cases a lot of changes are occurring in a flower, mainly the plant body. So now seed coat, if you know what exactly is integument, I'll just draw it and explain. So these integuments are the ones which are turning into a seed coat. So that is the outer integument is turning, that we call as a testa, and there is a inner integument. So inner integument is called as a tegmen. Okay, so we'll see what exactly is this. I'll just draw you the structure of the ovule that is there in the ovary. So let's see that, and then I can explain you this. So now this is the ovary. Okay, so ovary is placed onto what? So ovary is placed onto the thalamus. Okay, so this is the thalamus or the receptacle. Then what do you see inside that? So inside the ovary there is a ovule, which is actually, I'm just drawing an inverted ovule here. So you can just see it like this. So this one is a ovule now having a opening here as such. Okay, so this is what, this part is ovary. So I'll just label this. So this one is ovary, and this part what you see, so this is basically a ovule. All right, now this stalk is there, no, so that is basically joining the ovary, so that is a placenta will be here. So that is a group of tissue are there, so that group of tissue in the ovary is holding that ovule. Okay, now here, so there are coverings, so there directly, this is not the structure, so this is a covering called as integuments you can find. So now you know I'm just drawing one line here, right? So these are called as integuments. So these integuments are the ones which are forming the testa and tegmen in the seed. Okay, so what exactly happened, as you already have studied in class 7, class 6, so this concept like you know it is repeated. So you know that the fruit is basically a ripened ovary, right? So seed is basically a ripened ovule. So what is the meaning of that? So that is because the ovary is turning into a fruit later on. All right, so that is after the fertilization, and ovule is turning into what? Ovule is turning into a seed. So now this is the seed that we are talking about. So here these seed coverings, what you see, it is not only till here. So you know it will be till this end, that is chalaza end also. I'll just explain you what is that. So now you know this whole thing will be covered. So that means you can see this being the outer integument. So this part. So there is one more that we need to draw like this, because there are two coverings. Okay, so one is outer covering, there is inner covering. So this one is the inner covering. So this way it will be there. Okay, so this is the outer integument which is becoming a seed coat. Okay, so this is becoming a seed coat, and here you see, so this is just the outer part. So this whole part is basically outer part. Then you see this part, so this is basically a inner integument. Okay, so what is inner integument? So that is on the inside, the lining, the covering is there for a ovule. So this is a inner integument. So now what exactly will be there inside? What exactly is happening? Any idea? Any of you? So inside this, no, I'll just label what are these different parts are there. Okay, I'll just label this so that you will understand what exactly is forming a seed coat, what exactly is forming a cotyledon, and what exactly is forming a embryonal axis. So we'll see that. Okay, now inside this structure what you see is, so there is a structure called as a embryo sac. Okay, so now this part is basically a embryo sac, and this embryo sac, when we mention, so this is having a antipodal cells here. Okay, so there are antipodal cells that you see, that is towards the opposite end of the micropyle. So this end where you see a opening, so this opening is called as a micropyle. Okay, so this is called as a micropyle. So now you know towards the micropylar end you can see a egg apparatus. So that is egg along with the synergid. So they are having their own nuclei. Antipodal is also having its own nuclei, and here you can find a polar nuclei. So that means now this is the whole structure where you can see formation of a seed coat soon after the fertilization. So in that case, this outer and the inner integument. So outer integument after the fertilization. So this is gonna become the testa. Alright. The inner integument is soon after the fertilization. So this is gonna become the tegmen. So that means we call that. So it is basically a covering, protective shield for this embryo which will be there inside. So where is the embryo then? So here we were discussing, so there is a embryonal axis that is present. So where exactly this embryonal axis you can see? So that is inside this. So protective covering testa and tegmen are formed. So it is protecting what? It is protecting a embryo which is growing inside here. So this whole cell, I'll just show you this. Now after this triple fusion, so this whole part what you see, what which I'm labeling in a green, so I'll just highlight that. So just a second. Yeah, this whole part, just a second. Yeah. So you see this whole part, right? So this whole part is gonna become the endosperm. Okay, so that is after the triple fusion. So this will convert itself into a endosperm of the seed, and now you see there is this part remaining, right? So that part is where you can see a zygote or an embryo. So I'll just change the color for that, so that you can understand. So now this part what you see here, so this is gonna become the embryo. So that is zygote is formed. So zygote is converting into an embryo. So that is the seed embryo. So now when we mention about the seed having a specific structure, so seed structure, just a second. Yeah, so seed, when we mention, so it is having a seed coat, that is testa and tegmen, which is formed after the fertilization. A protective covering will be there. Then you know the outer integument is the testa and the inner integument is the tegmen. Okay, and other than that what you see here is a cotyledon. So what exactly is cotyledon? Here I was just drawing and explaining. Now I told you that this is gonna be a endosperm. So let me just change the color so that you can see. So I told you that this is gonna be a endosperm. So endosperm you can find where? So more of an endosperm you can find mainly in monocots. Okay. So that means the cotyledon will be papery cotyledon. So in monocots when we mention, the cotyledon is actually a papery cotyledon that you can find. Alright. And you know rest of it is basically here you can find the embryo. So this is gonna be a embryo. So that means you can mainly see endosperm as the nutritive supply for the development of the embryo, where the embryo is later on going to give rise to a plumule and then a radicle. All right, so plumule will form what? Plumule is going to form a shoot system. So this is going to form a shoot system, and you see there is a radicle. So radicle is going to form a root system. Okay, so you can see different changes that are going to occur in a seed structure. So that is after the fertilization. So lot of changes will be there. So that embryo is the one which is having a main capacity where it can germinate, it can form a plumule, it can form a radicle. So plumule, once it is generated, it will develop a shoot system, and the radicle, once it is formed, so it is going to develop the root system. Okay, so this is only after fertilization. But you know, so you might have heard in certain cases, like you know, even without fertilization, so there is a seed formation. So there are different processes that are there. So that is only because of the hormonal changes that are occurring in a plant body, or you know, in horticulture industries. So what they usually do is, so they'll apply these hormones to the fruit, I mean not fruit, the ovary. Okay, so in that case, because of the hormonal changes, even without the fertilization, you can see a seed formation. But you know, lot of cases, these are not viable seeds. Okay, so that is, they are not a living seed, so they are of no use. But here what you see, the one which is developed only after the natural fertilization process that occurs, so they are a viable seeds, or they are a living seeds. But rest of the cases when we discuss about, so those are non-viable. So why do we say non-viable? Because you know, the fertilization has not occurred naturally. What they have done is, they have injected the hormone in the horticulture industry, agriculture industry. So basically, you know, to sell the fruit, or maybe to get a fruit in a faster rate, so they'll inject a hormone to these fruits, and basically naturally, even without fertilization, you can see a fruit formation and seed formation. But the seeds are not viable in such cases. So this is, you know, the seeds are not viable when the hormones are applied, or otherwise after the fertilization, so they are a viable seeds. So let's have a look at this. Are you all understanding? So now here, as you can see, so we discussed about the embryonal axis and the cotyledon. So I'll just show you this diagram. So this is the dicot seed. Okay, So this is which seed? So this is actually a dicot seed. Why do we call this is a dicot seed? Any of you? Why is it a dicot? What is why we say it is a di? Di is what? Di is two. Okay. Cot is basically two cotyledons are there, and hence the name dicot it is. Okay. So two cotyledon. Where are those two cotyledon? Where is endosperm then? Like all the endosperm which was there in a seed, so that has been used up in converting this endosperm into a cotyledon. That is, for the formation of cotyledon, all the endosperm, that is nourishment which is provided for the growth of the embryo, so all that has been used up, and that is how we see a cotyledon there. So there is no endosperm, so that is why we call it as a ex-albuminous or non-endospermic or ex-endospermic. So what is the meaning of that? Basically there is no endosperm formation. So even though endosperm was there, all that endosperm is converted into cotyledon. So all the nourishment is used up in the formation of two of the cotyledons, and hence it is a dicot. So two cotyledons are there, so without the endosperm. So what about a monocot then? Monocot is having a lot of endosperm. So if you see a maize seed, so I'll just draw it here and explain. So this is a maize seed. Okay. So in maize, what exactly happens? So that is bhutta, what you eat. Okay. So in that case, what exactly happens is, so this is a maize seed with the slightest of opening that you can find over here. All right. Now, there is no cotyledon here, like, easily distinctive cotyledon is not seen, but a small, so there is a separation over here. So this separation is basically for the endosperm itself. So this whole part is actually a endosperm, and you see this part here, right? So this part you can find a thin one. So thin, we can say a papery like, so not just thin. Okay. So we can use the term papery like cotyledon, you can see just like this. Okay. So this papery like cotyledon is present, and hence we call this as a monocot. So there are no two cotyledons, but there is only one cotyledon, and hence the name monocot it is. It is not a dicot. Here this is a dicot because, so two big cotyledons you can see. So this is one cotyledon, this is one cotyledon, and here you can see a embryonal axis, but this is not the case with respect to maize. Alright. So in maize, what is the condition? So endosperm is there. So this endosperm is basically, you can see this is a nutritive tissue, and here a starch layer will be there. So there is one more covering as such. So this is basically proteinaceous covering will be there for this. So this one on the outside, it is a proteinaceous covering, and here you can find the embryo. Okay, so that is the embryonal axis. So let me just change the color. Yeah. So this one is the embryonal axis, which is gonna develop into a radicle and a plumule. So in that case, this part is covered by a protective covering. So that is the radical part is covered by a protective covering. So that we call as a coleorhiza. Okay, so this one is called as a coleorhiza. So that is a covering for a radicle. Why, how do we say that it is rhiza? So one more term I'll just explain you so that you can clearly remember these terms. Okay. So now this is the papery cotyledon that you can find, and below you can find the embryonal axis. So there is a protective covering for this as well. So that is for the plumule, and that we call as a coleoptile. So this one is what? So this is a coleoptile. So there are coverings that you can find in a maize seed. So these maize seeds, so basically endosperm is found, then there is a papery cotyledon is found. So since one cotyledon that you can find, so it can be a monocot here. So let's have a look at this. I'll just explain you what is epicotyle, hypocotyle. So you can just see here, this part they are showing this is a hypocotyle, and here it is shown it is the epicotyle. So what exactly is epicotyle or hypocotyle? I'll just draw it here and explain. One second. Okay, so now let's say this is the embryonal axis. Okay, so I'm just drawing it straight away like this. Okay, so now here you can see a plumule formation there. So at the top, so let's say this is going to give rise to a shoot. So basically you can see a plumule formation. So this is where this part what you see, so this is going to give a root system. So this will give a shoot system. Okay, so this is shoot system, so which is basically a we can say a plumule is the one which is forming this. One second, so it is not writing, just a second. Yep. All right, so in that case this can be the plumule. Yeah. Okay, now this is the one which is going to give to a give rise to a root system. All right, so this, let's say this is going to give rise to a root system. So in that case radical is the one which is going to form the root here. Okay, so plumule is the one which is going to form the shoot in the other case. Now attachment will be there. So let's say this is a cotyledon is where it is attached. So let's say something like this, the cotyledons are attached over here. Now between this embryonal node and then a plumule, so you see this region over here, so this region can be mentioned as the epicotyle region. All right, and you see this region between the radicle and the embryonal node or axis. So this region, what is there? So this can be called as a hypocotyle. So that is below the cotyledon and just above the cotyledon. So you can find the structure. So this is about the epicotyle and a hypocotyle. Any doubt till here? Any of you? Just a second, I'll drink some water. Okay, so oh, and now I'm able to see your chats. I'm really sorry, I don't know why it was not visible. Hi Sushant, because it has two cotyledons. Yes, yes, okay. All this while that's what I was thinking, why there is no one. Okay, now it is visible. All right. Okay, so now here in this case the seed is attached. Just a second. Yeah, so the seed is attached to the stalk by the hilum. So what is that now? I'll just explain you here. Just a second. So how can you see attachment of a seed? So now let's say this is a seed now, all right, which is attached at the ovary. So in that case, so you can see ovule is the one which is actually turning into a seed after the fertilization. So there you can find the integuments as such. So this one is a micropylar opening. So this part is basically a micropyle here. So this part is basically a chalaza opening. And what exactly is the hilum? So as it is written here, the seed is attached to the stalk. So that is by the hilum. So where is the hilum? So now this is a stalk. Okay, and this can also be called as a funicle. Now you see this contact here, right? So between the ovule and between the stalk, so you see this common junction. Okay, so this we call as a hilum actually. Okay, so what is written here? The seed is attached to the stalk, so that is by the hilum, so which is a common junction. So now as the seed reaches the maturity, so this is going to lose the water. So what happens if the water is not lost? So if the water is not lost, basically it will get degraded. So just imagine eating a raw corn. So this will be very like it will be soft. But if you just keep it open for a longer time period, so there are bacterial growth, the fungal growth mainly you can find, so that can happen. So if it has lost the water content as it has reached the maturity, so there you can find maybe about 10 to 15% moisture can be there. Okay, so it will be there, but rest of the water is all gone. Okay, so in that case, at maturity, when it is dry, so when the seed is dry, so you can find that there is up to 10 to 15% moisture that can be there. Then it reaches the seed dormancy. So what is that seed dormancy? Anybody here? Anybody here knows like what exactly is seed dormancy? No idea about the seed dormancy? So we say that the seed is in a dormant condition. So what exactly seed dormancy is? Just a second. Yeah. So seed dormancy is basically, one second, when the cells or we can say enzymes. Okay, so now seed is there, so inside these seeds, so there are lots of enzymes which have remained inactive. Okay, so there are a lot of enzymes that have remained inactive, and we don't want those enzymes to remain inactive. So in that case, suitable condition is provided so that the enzymes get activated. So now it reaches the seed dormancy. So when is it reaching a seed dormancy? So that is basically when there is a seed is maturing and it is not undergoing any processes, like germination is not occurring as the suitable conditions are not there. Okay, so as it reaches the seed dormancy, so there is this particular seed is arrested at a particular stage. So now the micropyle is basically the opening. So what is that opening? So now if you observe a seed structure, so I'll just draw it here itself. Yeah, so if you observe the seed structure, all right, so let's say this is a seed now. So on top you can see a scar on the seed. Okay, so this is a seed and you see a scar on the seed. Okay, so something, so that is basically a micropylar opening. So this is a micropyle opening itself. So through this micropyle opening, what happens? So there is an entry of oxygen inside, so there is an entry of water inside, so lot of changes are gonna occur. So why the seed is in a dormant condition, that is inactive condition, is because it has not got the suitable conditions for the germination of a seed. But when a suitable condition is provided, that means it is getting a proper amount of oxygen, it is getting a proper amount of carbon dioxide for photosynthesis, or it is getting a water, suitable temperature is provided. Okay, so this is all required. So temperature, then the oxygen concentration, carbon dioxide concentration for photosynthesis, then water is required. So different conditions are required, so only based on that the seed will undergo germination. If the suitable conditions are not there, so seed does not germinate easily. Okay, so here formation of fruits when we mention, so this is mainly after the fertilization process itself. Okay, so cell division in the ovary, so that is after the fertilization forms fruit. So this is basically the ovary is the one that is turning into a fruit here. Okay, so wall of the ovary forms the pericarp. So in that case, so as the wall of the fruit, so that is the outermost covering. The role of the pericarp is seed protection and the dispersal. So this is in case of what? So those plants which are not edible. So if you have seen them, so I'll just draw it here. So this is basically embryo sac that I need to draw inside this. So this, I hope you got this part. I'll just explain you how exactly this looks like, the pericarp with respect to that. One second, so I'll just explain this. Hi Shashant. So here I'll just explain you in this part, like only when there is, so you can see this part here. So wall of the ovary forms the pericarp. Okay, so that is the outer covering. So that is the wall of the fruit. All right. So now one more statement is given. The role of the pericarp is protection and the dispersal. So what is the meaning of that? So if you have seen this type of a pod, this is actually our fruit pod itself, which is not edible anyway for humans. All right, then you see many of the compartments there inside. All right, so many compartments will be there. So these many compartments, what are there? Yeah, so these compartments, what are there? So they'll have their own seats in them. Okay, so they'll have their own seats. This is about the seed that I'm talking about. So they'll have their own compartments. So this part is actually a ovary, okay, which is not an edible one, which has turned into a fruit, but non-edible, or we can say not edible one. All right, but this one can be, this is mainly the growth of this particular tree when we mention. So these will occur whenever there is a requirement. So whenever they want to continue their generation, so they'll be undergoing fertilization. So this is a ovary, and inside this what you see, so these are basically ovules itself, and ovules it is. Okay, one second. Yeah, so this is basically a ovule, and ovule, when we mention ovule, is the one that is forming a seed later on. All right, so in that case you can just imagine, so this particular fruit is there. So this is not the edible fruit, but you can see a covering. So what exactly does it do? It is doing a function of protection. So this does provide a protection. And if you have walked in gardens and all, or maybe parks, so you can see lot of trees. So this type of fruit would have fallen, and you can see that the seed dispersal, like, the seeds will get spread here and there, and again wherever the seeds are falling, so there is a growth of a tree. Okay, so in that case you can see this one, the role of the pericarp. So that is the fruit wall. Okay, so role of the pericarp, that is mainly the fruit wall. Just a second. Yeah, so this is mainly there for the protection as well as the dispersal. So this is the dispersal, where they will get dispersed and they'll fall off, and again there is a continuity of their generation. So this is what I was talking about the pericarp. So that is with respect to the fruits. All right, so this is with respect to fruit. Now the fruit wall, so this one is a tender coconut. What is this? This one is tender coconut. So in tender coconut, what do you see? So basically there is a pericarp. Like, pericarp is again divided further, where you can see a epicarp, there is a mesocarp, and then there is a endocarp. Okay, so these all these changes you can see only after the fertilization has occurred. So these are the parts in that. Okay, so there is a epicarp, mesocarp, and endocarp. So epi being the outer one, so meso being the middle one. Okay, and endo being the inner covering. So this one is inner covering. Then what is that? So this is the endosperm. So what does the endosperm do? So endosperm does the function of nourishment, so providing a nourishment for the development of embryo. So where exactly is embryo? Here it is labeled as an embryo. So have you seen a coconut embryo? Anybody? So if you have seen, whenever you open a coconut, okay, so at the bottom you can see a small whitish dot. So somewhat like this. So let's say this is the center now. So in the center you will see some small whitish dot, like, not dot like, so somewhat dot is, again, it is too small. So we can say somewhat a pepper sized embryo you can find. So that embryo is the one which will start to germinate. So that is for the production of a plumule, and also one second, so this is for the formation of a plumule and also for the formation of the radicle. So in that case, you can see this embryo is the one which is way too important, because embryo is the one which is forming a shoot system, which is forming the root system. So in that case, who has to provide a nourishment to the developing embryo? So that is the endosperm. Endosperm is the one which is providing a nourishment to the developing embryo. So any idea how the water gets inside a coconut? Any of you? How the coconut water gets inside, or how exactly this tender coconut is sweet? Micropyle. So this is in the tree. So how exactly this is absorbing the water? Micropyle. So that is, tender coconut is still in the coconut tree, and we just remove it from the tree, right? So in that case, mainly osmosis process is where you can see the water getting inside, and that is through the xylem. So water formation is there, and this tender coconut is sweet mainly because there is a phloem. Okay, so phloem is there, and phloem is supplying as the photosynthesis occurs. So there is a glucose supply that is there, and hence it is sweeter. Okay, so based on a different conditions, you will study about this, like how exactly water gets. So that is mainly through the osmosis process, and then into the xylem the water goes, and from the xylem, this is reaching this part. So that is mainly the coconut part. So that can be because of the water absorption process. So that you will study later on. So this we discussed about the ovarian wall. So that is being the pericarp. So this was about the pericarp, the outermost wall, and which is protecting the seed. So that is there, and other than that, the seed dispersal also it is gonna help in. Alright, so micropyle is going to help in water absorption. So that is in a dry seed. So this is about the fruit I was asking. So now when we mention about a seed, what you said, Shushan, so that is right. So the water absorption can occur through the micropyle, the oxygen intake can occur through the micropyle. So before the fertilization, the pollen tube will enter through the micropyle. So this opening is of lot of use for the plant body. So mainly with respect to seed formation or for the fertilization process. So here in this case, the fruit protects the seed and helps in the seed dispersal. So same thing with respect to what we saw for the pericarp. Alright. Now seed is a ripened ovule. So this is a ripened. Why are we using the term ripened here? Every time we use, the fruit is said as a ripened ovary. The seed is said as the ripened ovule. So ripened is basically after the fertilization, not by injecting some hormones. So we do it since lot of development in technology is there right now. So we inject a hormone, even without fertilization, so immediately we are getting a fruit. So not by all that method. So now when we mention a ripened ovule, so this is a small eye like structure. Okay, not eye, it is the embryo. Okay, so this part is the embryo in a groundnut or a peanut. Alright, so in that case, the embryo is the one. So if this is there and you are soaking, basically you are soaking it in a water, then sowing this peanut seed in a soil. So this embryo is the one which will start to germinate. It will form a shoot system, okay, and the same embryo itself, so that will have a radicle. So same embryo is going to form a root system, and that is how you can see a growth of a plant. So that is by absorbing the water through the root, and as the photosynthesis occurs, the food is also supplied. So why is the seed so important? The seed is important because it can continue the generation, plant generation after generation, to maintain the species level, so that they do not go extinct. So there are a lot of animals who have gone extinct. There are a lot of plants that they have gone extinct. So all this shouldn't happen. So there is a continuity in their generation, so we need to get different we can, like mainly the plant, if we are saying, so we'll talk only about the plant. So there might be a case that this plant might be of a medicinal value for a human being, or it can be not just, let's not look on point of a human purpose, but for the continuity of their generation. So in that case, they'll always make sure that the pollination is occurring, fertilization is occurring. So even if the pollination is not occurring, so they'll have this clistogamous flower, so that is a closed flower will be there. So with that closed flower, so they'll anyway make sure that the seed set is formed, that is seed is being formed. So that is how they'll make sure that there is a seed formation, and they'll continue the generation as they'll continue as they'll have a embryo. So now grain is what then? So just now we were discussing, there is a pericarp, that is a fruit wall, and fruit wall and the seed coat. So let's imagine this is a seed coat, that is integument, the outer integument, the inner integument, which we call as a testa and the tegmen. Okay, so this testa and tegmen have been fused. Okay, so there is no separation between them. So this testa and tegmen are completely fused. Okay, so this is a fused structure, and this fused structure is forming this grain like structure. So this is now a grain having the endosperm inside. Okay, so there is a endosperm. So this endosperm does the function of providing a nourishment. So where is the embryo then? So in that case, you can see embryo that can be here at this part. Okay, so there is a embryo here, and this whole thing is basically a endosperm. So let me just label this. So this one is a endosperm, and here this is the embryo. All right, so both of these are being produced. All right, so this is why is it produced? So that is to continue their generation after generation. So in that case, the testa and the tegmen are anyway fused along with the pericarp, that is the ovarian wall. So all of this together, like we see in case of maize, like we see in case of wheat. Okay, so this is the fruit in which the fruit wall and the seed coat are fused together to form a protective layer. So there is no fruit separately, there is no seed coat separately, so both of them are fused, and hence we call this as a grain. So there is a difference between grain and a seed. So what is that difference? So seed, there is no fusion, but in the grain you can see that the fusion between the pericarp and the seed coat that has occurred. Any doubt? Is this clear? Alright. So dicot embryo is having one embryonal axis, that is two cotyledons will be there. So there is a epicotyle, which I told you, like the axis, it is basically the part of the axis above the level of cotyledons. So in the previous one here also, so you can see now, so this is this part basically you can see above the level of a cotyledon what you find. So there you can find the epicotyle region. Okay, so this could be the epicotyle region. All right, so this one is the epicotyle region. So now between the radicle what you find, so that is between the radicle and the embryonal, in the embryonal axis, but the embryonal node, so that could be called as a hypocotyle. So this can be, okay, not here, so let's see here somewhere. Okay, so not this labeling, so I'll just erase this. 1 second, I'll just label it properly. Yeah, so now do you see this part, right? So this part could be this hypocotyle region. Yeah, so this part could be the hypocotyle region there. So this is the embryonal node that might be there, and this is forming a shoot system. So now monocot embryo, 1 second, so let me just check this. So here this one I was just discussing, so terminates in a plumule or a stem tip. So plumule will give rise to a shoot system. Hypocotyle is the axis, is the part of the axis below the level of the cotyledons, terminates in a root tip. So basically root tip formation, shoot tip formation can occur as the epicotyle and the hypocotyle are going to push through the ground. So now if you remember epigeal type of germination, okay, and then there is a hypogeal germination that you can find. So in epigeal or hypogeal, so what is going to happen? So in hypogeal, the seed will remain down itself, all right, but this particular shoot system has to push above the level of ground and it has to come out as such, okay. So if I just explain it to you this way, so let's say this is the green part, that is just shoot system. So this one has to push above the soil level and it has to form a plant body. So in that case the seed has remained down itself, so that could be the hypo one, okay. So now based on a condition, hypocotyle is the part of the axis, so that is below the level of cotyledons. So this is going to terminate in the root tip and then it is covered by a root cap. So there you can find a root cap as well. Now when we mention about the monocot embryo, just a second. So now as we mentioned, so now monocot embryo has single cotyledon. So that I told you, like papery cotyledon will be there. So this is a papery cotyledon. Alright. So that papery cotyledon is called as what? So the papery cotyledon in case of a monocot embryo is called as a sketalum. Okay. So this is basically a sketalum. So monocot embryo has single cotyledon, that is at the end of the embryonal axis. The axis above the level of cotyledon is the epicotyle. So in that case the axis above the level of cotyledon, so that is, let's say this is the axis and this is the embryonal node and this is the plumule, this is the radicle. So in that case this could be the epicotyle and this could be the hypocotyle. Alright. So in that case the epicotyle consists of the shoot apex and some leaf primordia, then which is enclosed in a hollow folia structure, so that is a coleoptile. So now this one, if I say this is a plumule, so there is a covering of a sheet, okay. So the sheet covering which is there, so that we call as a coleoptile. So this is a hollow folia structure it is. Now the axis below the level of cotyledon is having the radicle, so that is with the root cap, and that is basically protected by another sheet, so that we call as a coleorhiza, okay. So this one is a coleoptile. I think you remember, since I wrote about this one in the very beginning of the session. So this one is the coleorhiza. So these are the different parts that you can find with respect to these seeds, okay. Just a second. Yeah. So this is a wheat monocot. So because you can see there is a protective covering. So there is a plumule and the radicle, and here this is a endosperm. There you can find a single cotyledon that will be there, okay. So here it is a testa, then there is the micropyle is where the oxygen can be taken in, the water can be taken in, so that is for the germination process. So this one is hilum, is just a junction where you can see the ovule and the stalk of the ovule are attached, okay. Now this is the open structure which I had added in the very beginning. So this I'll explain you. So now you can see there is a aileron layer which is a proteinaceous layer. So this is a protein containing layer. So here you can see this part, just a second. Yeah. So this part is basically a endosperm part. Alright. So that is the endosperm part. Then you can see a epithelial tissue lining that is there. Then there is a sketalum or a cotyledon. So what is that sketalum or a cotyledon? So this is actually a a papery cotyledon that can be found. So this papery cotyledon is called as a sketalum as well. Hi Ria, good evening. All right. So then, covering the plumule, you can find a coleoptile. Covering the radicle, you can find a coleorhiza. So whole of this is basically a embryo. So this is the longitudinal section that has been shown here. So this is a longitudinal one. So this is position of plumule is shown. Sketalum is shown here. Then other than that, the remains of the style is shown. So this is, you need not worry. The endosperm and position of radical. So all that is correctly shown. So that while you are drawing, so if in your school if they ask you to draw, so maybe you can go ahead with this diagram. Okay, so because all the labelings are there in the other one. Okay, so development of a seed, so this is only after the ovule gets fertilized. So this I told you, so the based on developmental factors, or if the endosperm is present, so that we call as a albuminous seed. So if the endosperm is not present, so that we call as a non-albuminous seed. Okay, so based on the number of cotyledons, so if one cotyledon is there, so it is a monocot. If two cotyledons are there, so it is a dicot. So now if remains of the nucellus are present, so that is called as a perisperm. So this is, you do not have to take that into mind. So that is basically nucellus is forming a perisperm, which is the content that is present in and around the embryo sac. So if you remember this structure again, so there is a ovule now. Okay, so this one is a ovule, and here you can see a embryo sac. So here in this part you can see a embryo sac. So what is mentioned in here? The remains of the nucellus. So surrounding this structure, what happened? Yeah. So surrounding this structure, so there are certain cells. All right. So those cells are called as nucellus. So what is this saying about the nucellus? So that is going to form the perisperm around the other cells. Okay, so sketalum is basically, it is a papery cotyledon that you can find in a monocot seed. So let's say this is a maize seed now. Okay, so there is an opening, so that is always there. So this lighter opening that will be there at this part. Okay, now there is a separation with the epidermal layer. So now here you can find, so this whole part, if I have to just mention, so now this part, like this whole part, so let's just take it as such. So this whole part is called as a sketalum. So this is actually a papery cotyledon. It is like papery we are saying because it is too thin. Okay, so this is thin, but this is a cotyledon that you can find in case of the maize, that is butta, what you eat. Okay, so what will be there? So when you eat it, so basically you can see some whitish structure that is still remaining. So that is the sketalum actually, which has still remained. So that is a papery or a thin cotyledon that is there. Is that clear, Ria? So here anyway, as usual, so there is a radicle, then there is a plumule, okay, so which will give rise to a root system and a shoot system there. So this one outer covering is basically, there is a one more covering, so that one more covering or the outer covering which is there. So this is called the aleurone layer, which is actually a proteinaceous layer. So let me just complete this. So this way it will be there, okay. So here the seed is a developed ovule, fertilized and developed ovule, developed ovary, fertilized and developed ovary. Which option is right here? Thank you, Ria. Any idea which option is right? So this seed is a fertilized and a developed ovule. Okay. So this is a fertilized and developed ovule. So only after the fertilization you can see a ovule formation. I mean ovule that is converting into seed. So seed formation or after the fertilization, and hence it is a developed ovule. Now the tegmen of seed develops from, so there is a testa and tegmen that I told. So tegmen development, how do you see? So tegmen development is basically occurring from perisperm, funicle, inner integument, outer integument. Any idea on this one? Okay. So this one testa is for a outer integument. The tegmen development is from the inner integument. So this is the answer for this one. Okay. So sketalum is endosperm or papery cotyledon of a monocot, protective covering of the radicle and protective covering of the plumule. So what is sketalum? No idea? So sketalum is basically a papery cotyledon of monocot. So this one is a papery cotyledon. So that is a thin cotyledon that you can find in case of monocot. So that is a papery cotyledon. So protective layer of a radicle during the seed germination. So that is for the radicle. So it is a coleorhiza. So if it was asked for a plumule, so it is a coleoptile. So you can just remember using radicle, rhiza, plumule, and that is mainly for a ptile. That is coleoptile. Okay. So X albuminous seed, the food is stored in. So this is X albuminous. That means all the endosperm is used up in formation of cotyledon. So only in the cotyledon you can see a food storage that is done. So here in albuminous seed, the food is stored in dash, and non-albuminous seed it is stored in dash. So in albuminous seed where it is stored in the endosperm, and in non-albuminous seed it is stored in the cotyledon. So this is the one. Alright. Okay, so before I end this session, so let me just say about the batch that we have. So this is the batch in English. So for ICSE class 9, so this is Guardians batch, which is a all subject batch, started on 1st of June. So maths, physics, chemistry and bio will be covered. So if you are subscribing for class 9, so you will be getting class 10, 11, 12 subscription absolutely free along with this. Like, meanwhile you can prepare in your free time. You can prepare for other classes as well. So introducing community. So never miss on any update from your favorite educator. Get session notes and practice material. Share your feedback directly with the educator via polls. Check out the link to the educator community below. So you can just find the link to the community in the description box. So opportunity for all learners to report any inappropriate content in the video. So be the first one to report a particular issue to claim your prize. Report any inappropriate content using the form in the description. Alright. Okay, so I have one more session to go. So I'll just end this session. So thank you all. So see you all in the next session. Subscribe to our channel, that is Unacademy, ICSE and ISC boards. Click on bell icon to get notified about these sessions. Like and share the video and download the Unacademy Learners app. Now, so this PDF if you need, basically you can join my community. So the community link you will find in the description box, and there I'll be sending it. Okay. All right. Bye bye everyone. Take care. Thank you so much.